Welcome back. We are going to continue our study of the cell. Last time we looked at the structure of the plasma membrane, and this time we're going to look at the concept of membrane transport. We're going to look at how that membrane functions to allow things in and out of the cell. So the membrane is what's called selectively permeable. Some things it's very permeable to, like gases and anything that's super small, uncharged, some polar molecules, they can basically go right through. Lipids like lipids, so if you're lipid soluble, right through the lipid bilayer. Other things, charged polar molecules and ions, there's almost zero permeability to. So it's selectively permeable. Some things, open door. Other things, not open to it at all. We can divide how things cross the membrane into what are called passive processes and active processes. These have to do with the input of energy. Passive processes do not require energy. Active processes do. So we're going to start with passive. When we talk about passive and active, we talk about gradients. So in terms of membrane transport, it's usually a concentration gradient, but a gradient is going to exist in many forms as we go through the body. We're going to see pressure gradients later, but here we're talking about concentration gradients. How much of something is on one side of the membrane versus the other. Whenever you go from high to low, you're going downhill. You're going down your gradient. This does not require energy. This dude just gets in the wagon and boom, it goes. He didn't have to put in any energy there. Whenever you go from low to high, it's going to require energy. Okay? So passive processes are going to go from an area of high to low. So this is what we're dealing with when we're talking about passive processes. This is what we're going to deal with active, where we would need to input energy. But we're going to start with passive. There are multiple things that are going to affect how efficient transport occurs. The first would be temperature. Increased temperature, molecules bump around, they move around, they bump into each other more often, and transport's going to happen faster. All of this relies on random molecular motion. So the more energy via heat we put into the system, the faster everything moves, and the more transport you're going to get. Molecular weight is going to play a role. Smaller moves faster. How steep the gradient is going to play a role. Steeper the gradient, faster the transport. The more surface area we have for transport, the faster the transport. And the more permeable the membrane is to whatever is moving, the faster the transport. Okay, so there's several different things that are going to play into this. So the simplest possible passive process is what's called filtration. You are using a physical pressure to force fluid and solutes through a selectively permeable membrane. If you've ever used one of these guys, you are familiar with this concept. So an old school coffee maker uses a filter paper, and that filter is going to function as your selectively permeable membrane. You put your coffee grounds into the filter, and then water is going to flow through them, and the physical pressure on water is gravity. So the force of gravity pushing on water forces it through the coffee grounds and then through the filter, leaving you with coffee. Similar in the body, okay, we're not going to use gravity. We're going to use what's called hydrostatic pressure of the blood or blood pressure. So the amount of pressure in the blood as it comes through these small capillaries is going to force water and solutes out of the blood and into the tissues. The rule is if you fit, you filter. It's all about a size principle. If you can fit through the clefts, then you're going to filter out. If you can't, you won't. So look at the size of this red blood cell here. We should not be filtering red blood cells. But water and really small solutes, sodium or potassium, something like that, yes, they are going to be able to be pushed out due to the pressure in the blood. So filtration is very unexact. If you fit, you filter. Diffusion comes in a few different varieties, but it's always going to follow that movement from high to low. So we're going from an area of high concentration of a solute to an area of low concentration. The simplest form is just simple diffusion of a lipid-soluble solute across the membrane directly. It's a high concentration here, a low concentration here. These guys can diffuse through the lipid bilayer because they're lipid-soluble. They will move to equilibrium. If your solute isn't lipid-soluble, then you're going to need to use something called facilitated diffusion. Here we're going to use some of our proteins that are embedded in the membrane. So we're going to have a carrier or a channel. Carriers are generally more specific than channels. And it is still going to be going from an area of high to an area of low. So this is still diffusion, going from high to low. We just need a helper. We need a facilitator in the form of a protein to get our solute across the membrane. The third type of diffusion involves the diffusion of water instead of a solute, and this is called osmosis. So osmosis is a form of facilitated diffusion. It uses a channel called an aquaporin to increase the permeability of the membrane. Some water can diffuse through on its own, but aquaporins are going to make the movement of water a lot more efficient. So we're going to take a look at osmosis because this is an important concept. The principles of diffusion, going from high to low, are going to come into play over and over and over and over and over and over and over again in this class. So you want to make sure that you understand diffusion and osmosis. So osmosis is the movement of water. So now we're not talking about the movement of a solute. We're talking about the movement of water. Water is going to move from an area of high water concentration, which also could be said an area of low solute concentration, and it's going to move to an area of low water concentration, also high solute. Okay, so when you look at this, you want to think water follows salt. Water will always move to wherever there is more solute. Water goes in and out of cell membranes constantly. So wherever there's more solute, so here we start side A, side B. Side B has solute, side A does not. Now, the membrane here is not permeable to the solute, so simple diffusion isn't going to help here. We can't get the solutes to equilibrium. So water will begin to move to side B in order to dilute out some of the concentration here. So water will move to wherever there is more salt. Whether that is in or out of a cell depends on the cell and the extracellular environment that it is sitting in. And that's where the concept of tonicity comes in, or the effect of osmosis, essentially. When we think about the fact that all cell membranes are permeable to water, we really have to think, wow, we have to manage how water moves, because water will go to wherever there is more solute. Most of the time, we want our cells to be in what's called isotonic solutions. We want the solution around the cell, the fluid around the cell, to have the same tonicity or solute concentration as the intracellular fluid. Basically, we want the extracellular and the intracellular fluid to match. Iso means same. So when a fluid is isotonic, it's going to have the same concentration as the intracellular fluid. So in the case of sodium chloride, our intracellular fluid is 0.9% sodium chloride. So our blood plasma, for example, which would be the extracellular fluid surrounding red blood cells, which you're looking at here, is 0.9% sodium chloride. Now, water is always moving in and out of these cells, but it's going at the same rate in and out because there's no draw one way or the other. There's not a gradient. Everything is isotonic. Let's say you get super dehydrated, like basically you're dead at this point, but we're going to use it as an example. Hypertonic solutions have more solute than the intracellular fluid. So the fluid around the cells is hypertonic to the cells. So we're using an example of 10% sodium chloride. Literally, you would be dead. Don't do this. But for our purposes of an example, when you put a cell in 10% sodium chloride, there is a huge draw on water. Water is going to move out of the cell, and it's going to have the effect of shrinking up the cell, and that could be devastating to the cell. Now, this is showing all water going out, but I do want to point out that because of random molecular motion, there is still some water going in to the cell. But by far, the overall net movement would be out. So you can imagine that if all of your cells begin to shrink up and they basically become like little raisins, that would be a bit of a problem in terms of function of the cell. Similarly, a hypotonic environment is also a bit of a problem. So here I'm giving you the example of distilled water, 0% sodium chloride. If you put a cell in there, suddenly the intracellular fluid is highly concentrated. Water is going to move to wherever there's more solutes, and it's going to keep increasing the size of this cell. In a red blood cell, you can actually blow them up like an overfilled water balloon. So when you think about this in terms of why we talk about homeostasis and maintaining stable internal environments, this is just another example of the effects. If suddenly your extracellular fluid gets very salty because you're dehydrated and you're losing water, there are effects on the cells. If suddenly we dilute out your extracellular fluid significantly, there are effects on cells. There's a reason when you go get an IV that you get something called normal saline, and it's 0.9% sodium chloride, because that's what's isotonic to your cells. We don't give you a distilled water IV because that's going to have a devastating effect on your cells. So when you think about this, and you think about the plasma membrane and the movement in and out, you need to think about the fact that everything has an effect. And when you go back to that basic concept that we talked about in the very first lecture of homeostasis, this is why. This is another example of why we have to maintain stable conditions. Now, all of that was passive. We were going from high to low. We didn't have to input any energy. The gradient itself drew the transport. Active processes require energy. So the first type we're going to talk about has to do with a gradient, but we're going in the opposite direction. We're going to go from low to high. We are pushing our wagon uphill. That requires energy. It takes energy to walk uphill. Okay. One of the main ones that we'll see in terms of these pumps, these energy-driven pumps, is the sodium-potassium pump, which is an example right here. This is an incredibly active pump. Huge amounts of your cellular energy go to running this pump. We're going to talk about it later in the muscular chapter, again in the nervous chapter. Multiple different physiological mechanisms rely on having sodium in high concentration in the extracellular fluid and potassium in high concentration in the intracellular fluid. The sodium-potassium pump consistently pumps both of those ions against their gradient, constantly takes sodium from inside the cell and pumps it outside where there's already a high amount of sodium. It takes potassium from the outside and pumps it inside where there's already a high amount of potassium. This maintains the gradients we need to run our physiological processes. This requires energy. So here's our cellular energy. ATP is coming into play here. So pumps are going to move solutes against their gradient by using the energy in ATP. So that's one type of active transport. The other type has to do with moving things through the cell in vesicles, membrane-bound vesicles. So when I want to bring something into the cell, it's called endocytosis. And there's two versions. Phagocytosis means that you're bringing in something solid, and only specialized cells can do it. Penocytosis, pretty much all cells do. Penocytosis is bringing in extracellular fluid and whatever solutes happen to be in it. So here you can see an example of phagocytosis. You have this particle, you're going to surround it, and because of the self-sealing quality of the plasma membrane, you end up with a membrane-bound vesicle called a phagosome. You're going to fuse that with a lysosome, which is going to chop up whatever happened to be in that particular particle. And so this is the process of bringing something in. Every single step requires ATP. So the formation of the vesicle, the movement of this vesicle from the plasma membrane to the lysosome, every single step requires ATP, which is why this is technically an active transport. It doesn't have to do with gradients so much anymore. It has to do with the fact that it requires ATP. Now, a really specialized case of this is called receptor-mediated endocytosis. Here, you actually have receptors, so remember that these would likely be proteins in the membrane, and when they bind their very specific ligand, it triggers the formation of the vesicle, again requiring significant amounts of ATP at each and every step. The opposite is called exocytosis, when we form a vesicle in the cell and then we want to release the contents into the extracellular fluid. Again, movement of the vesicle through the cell, the fusion with the membrane, all of this requires ATP, which is why this is active. Transcytosis combines endocytosis and exocytosis. So here what you're looking at is actually the cells that make up the wall of a capillary. And this would be your blood, and this is a muscle cell. So here you can see that there are vesicles that are moving in to this cell on the blood side, and then they fuse on the opposite side, releasing the contents to your muscle. So the hormones, insulin and glucagon, are going to use this method. So they basically use endocytosis on one side vesicular transport to get through, and then exocytosis on the other side. So it's a combination of the two different types of transport.